Hello, this is Elim Romero again. And right now, we are gonna do another downstream analysis. In this case, it's gonna be differential variability. I haven't introduced what that is. I'm gonna prepare a video where I'm gonna explain what that is in depth, what's the little theory behind, and how can help us. For now, I'm gonna type in SEG toolbox, and I'm gonna work on the same sample of the previous videos. So I'm gonna open the math file of CRC sample with no QC. And as before, we subsample here, selecting cells by class, and select cell type, alphabetical. Gonna take a fibroblast again. Sorry, something happened. It's take out deselect. Oh my god, what happened? Cell type? Alright, sorry, I had to stop the video. It was confusing because I need to do again the clusters. So those are properly clustered. So I'm gonna run again the SEE equals to latent clustering annotation, as in the previous video, with resolution 1, no highly variable genes, and annotate with the human database. Oh, sure. I got to double click on it, and that's gonna appear on the workspace. After it's in the workspace, I can call the function. It's gonna do the clustering, and after it finishes, I'm gonna look again at the fibroblasts. That's very important to have the classification properly. That's why I only pull it up fibroblast from one of the samples. Something that needs to be refined and verified all the time until you have something you feel confident. So in this case, it finished, and we're gonna do SEE. Well, now I'm gonna show you to open it in a different way, just SEE. And here you can do now import, and import from SEE from the workspace. Okay, and then that's selected, and now we should be able to look at the new cell types. This is what we got. And now we are going to subsample again, cell type, alphabetic, fibroblasts, select. Now I hope again to have, we have again the two different populations. So we have cell type, those are fibroblast, and batch ID, those are different samples. Great, we got the same as before. That's something we have to verify every time we do something, that we can replicate that anytime. So that is science, that we can do the very same steps and get to the same point. It might differ in some few cells, but overall looks similar to before. Now, what we are gonna do is differential variability, which is here, differential variability, DV analysis, or can be here in analyze and this differential variability here. We hit on it, we OK, use this, override if needed. Which species? This one is human, and we need to curate or clean the gene space, and that's the suggested. Then it's gonna do the that process, and then we're gonna do batch and select the A0 and the CRC. In this case, I had too few cells and filter everything, so it didn't work out. It's not gonna pass. But now the difference is that I can do the same, but instead of removing mitochondrial, it's gonna be no, removing some other, no, and no, and let's see if that helps us. That indicates that this sample is potentially low quality, so we gotta be careful. again, it failed. That means I will not be able to do differential variability within fibroblasts. Good test case scenario. I'll close this other. And okay, I'm gonna try to select something else and see if that can identify that. I'm gonna try enterocytes in this, enterocytes or endothelial cells, where those are. Let's try enterocytes, select, and unfortunately we don't have the two populations. It's just these few cells here. Then I'm gonna roll back to the test case scenario from this sample, sorry, from the toolbox. So I'm gonna type in again SEG toolbox and import the example from the data. Right, data from example. And again, we're gonna have to select a cell type that has in common within the two batches or samples. So we're gonna look at batches and potentially here in the middle, some here, some there. Let's look again at the cell type. Oh, what happened? Cell type. Okay, it looks like fibroblasts again could be a candidate, or alpha cells. I think those alpha cells. Let's verify samples, these two populations. Right, alpha cells we can take out. So let's select, in this case, alpha cells, and let's see how that goes. Okay, we have the two populations. Great. So we verify batch ID. We have the two samples. Cell type. Oh my God. Here. Cell type. Alpha cells. Okay, great. One way to verify, because all the time happens in with collaborators that, hey, why my two cell types are separated? Are those biologically batch effect, or those are technical batch effect? I mean, by sequencing or anything went wrong with the processing for sequencing or anything. So what we can do to verify that, it's UMAP again. Now that we have only those two cell types, so we can click on that. It's buggy. Let me close this other. I'm gonna hit and switch to 3D. No. It's this one. My bad. Is it next to it? Use existing. No. I want to do UMAP 2D. Yes. Override. Yes. And I want all the genes, because in this case, some few genes can be variant, and that's part of what we are going to analyze. So these two guys are still separating, which might mean something biological. Our task now is to analyze what makes them different. So spline-DV is a method to verify that. So we do differential variability. Use this. Yes. This is mouse. Remove everything you need, and it's gonna get me the differential variability within the two time points. And what I'm gonna do as well is the differential expression. Okay, this is what we got in differential variability. Let me, yeah, I'm done. I was closing something. Here we have two samples, and what to make them different we can explore here in this second. Select a gene from the sorted list. The list is sorted by the highly variable. As you can see, if we select at the second one, it shows the CTRB1 gene for the two samples. The above is 0.8 weeks and the bottom one is 14 weeks. And if we inspect that here, we can see that the orange is more separated from the lines than the blue one. The blue is within the curve. Means that the blue is stable, as we can see here in the expression level of the sample of 8 weeks. And if we look at this other CTRB1, we just need few cells to go crazy to identify that as variant. As you can see here, we have some high peaks, and that makes it different already. So what we do here, we consider these genes now as probably driving the effect of this. And this is the whole gene list that should be stored already. I need to look for it, so I'm gonna run it again because I'm not so sure where is that stored now. Meanwhile, let's do the E as well, so we can do something fun. We're gonna do that and store it there, and the 2 batches. Okay, we're gonna do 14 weeks versus 8 weeks all the time. The reference is gonna be the second all the time. Additional analysis, yes, why not? Yeah, those. I'm gonna stop it. Okay, it finished, and it's gonna get us the folder, and we just say okay, and here's the volcano plot again. Again, this gene, this other gene, and this other gene here and here should be in the main list of the analysis. KRT8, I know that guy. Okay, so we got that. We can just close this and open the spreadsheet and look at the upregulated. We're gonna see what happened to that sample at the 14 time point. The upregulated is gonna have that information. Now let me see if yeah, just wait me a second. I'm gonna look for where the spreadsheet for the differential variability is. All right, I'm back. I'm sorry. It says that it's gonna put it in the local temp SEG toolbox DB analysis is working there. So we're gonna go and look for it. It got me the DE and the temp as well. So we can go and look for the DB, which is below. Gonna hit on yes. It's gonna be mouse. Clean it. Compare the 2 time points. And then we get it. You want to explore? Yeah, sure. The most important is gonna be this table. And here I'm gonna put export Excel. And oh, it's letting me customize that. Great. So here it is, the 2 of them. Oh, my bad. So I can save it here locally. So I can do 0.8 8 weeks versus 14 weeks DB analysis. Just do it. Close it. I can close this now. So it wasn't safe here for now. So we open the DE analysis, and we're gonna look now at the DB analysis as well, which is located at the main position of the MATLAB directory. So it's gonna be this. And now we have DB. DB and DE together are powerful because DB can tell what is variant, but not in which direction. So one thing is that what we can do for having the analysis of DB, it's grab the top 100. We can do top 100. We grab that one top 100 and we're gonna look for enricher. Go in there, copy the gene list, and then we go to ontologies, biological processes, regulation of apoptotic process. Interesting, I guess. So we gotta be aware what the alpha cells functionality is and what these samples are about to learn about that. But look, it's consistent apoptotic neuron apoptotic process, regulation of apoptotic process, endothelial cell migration. Oh, my bad. If that happens, you can restore it by going back to another tab, coming back and just opening it again. Negative. Oh, it didn't. Actually, it didn't. Regulation of apoptotic process. No, it didn't come back. It's dynamic. Be aware of it. So we can come back to this table and positive regulation of apoptotic process. Good. The problem is that collaborators ask us which direction these genes are. You can see the gene list there, but if you want the table is export entries of the table. And you should be able to locate this file and open with Excel. Might not be easy. So what I can do is Excel and file, open, and I'm gonna browse for downloads and hey, wait a minute. It should be there, right? Let me just be sure. Not here. Downloads, it's there. I think then it's gonna be import data, and it's gonna be where was that? Import data. It was a tab here. Let me look for it. Gonna come back in a few minutes. All right, got it. So after you open wide Excel, just an empty Excel, so you're gonna go to data and then from text or CSV. Select that tab and it's gonna ask for some things. You can just load it for now and later we can worry about that. And it's similar to what Dr. Sai already downloaded in his process. In this case, what we can look is positive regulation of apoptotic process, this gene set. And one cool thing is that, for example, if we look for this FOXO3 expression here, we can come to this first button, I think, and then select the gene or paste. I'm gonna paste it. I wanna see the expression in these cells here. Just click on it, normalize input, yes. So I want A and B is gonna do library size normalization plus log1 plus. And should look like it. And as we see here, it's more variant. Oh, I'm sorry. Apply the same rotation to all tabs, not for now. So we see that is, no. Okay, we see this, and actually we see more activity in the left-hand side than in the right-hand side. So what this means is that FOXO3 is more variant into the 8 weeks. So it kind of suppresses and gets down the expression in the 14 weeks. So if we look for it in the tool here, we can go and look for FOXO3. FOXO. We just type FOXO3. Oh, it didn't look for it. Okay, let me look here in this other FOXO. Oh, it's not looking for it. Oh, yeah, wait a second. I'm having some difficulties here. We can do this and look for FOXO. FOXO should be somewhere here. Why is not looking for F? Well, we can do reverse engineering. Let's look for APO. Let's look for this APO, and it's more variant now in the blue than in the orange. That's in the 14 weeks. So we copy the gene name and we come back here to the sample. It's gonna be here. This button we select. Sorry, let's paste. For me, it's simple. APO, yes. That normalization, and then it's gonna bring this. Okay, interesting. What it's saying here is that if we look closely here, in the blue is more variant, and in the orange is more stable, more into the line, and that actually we can see that is widely active here, and we also see it here. It's more active in this top corner than in the left corner. If you see, it's wiggling a lot, so the red dots means it's turning on strongly, and it's just wiggling around. And we see more orange here. That means a stronger expression. That's why we see a lot of these expressions like wiggling, but above the 0 line. So if we come back here, we can realize that again, the 0-8 week is more variant, but in the 14 week is stronger the signal. That's how the spline-DV is developed. So we can look at these changes. Hey, it's something that should be like this or not. It depends on the sample and the biological context. But you have something and then you have something else. That means it's stabilizing, but stabilizing is stronger in the 14 weeks than in the 8 weeks. So that's something cool and might tell something. Another powerful way to look at this is we get the two. I'm gonna close the enrichment analysis and gonna come back to DB and DE. So a powerful way I found out so far, it's copying this spline-DV top 100 and going to find the overlap of those. I usually do Benny. You click on Benny and you can type in here DB gene set and copy that. And then we can bring the upregulated and see which ones overlap, if any. And we can bring also the top 100 and let's see if there is any overlap. And that's gonna be already highly variable and differentially expressed, upregulated in the sample. So here it's 81 only DB, 81 only DE, and 19 overlapping. Those might be really interesting. If I do copy this and do enricher of that, we might get some cool insights of what is happening. Autologous, and those genes are in endothelial cell migration regulation, angiogenesis, inflammatory response. Cool. That's very cool. So we go back and verify these genes are actually or not variant, and in which direction. Let's look for CXCL10. I know that's an inflammatory gene. That's what I want to see it, and I saw the inflammatory pathway. So if we look that here, CXCL10, we are seeing that it's more variant in the orange. Okay, that's changing. But in the expression level, we have to come back here and paste it. Yes, normalize A and B. And there you go. It's upregulated in the 14 weeks, and it's variant. It's changing quite a bit everywhere. So that's great. That's powerful enough already, and it's screaming something. It's screaming that at the 14 weeks is inflammation activation, what exactly we saw in the pathways. Great. This is one way to look at that, and another way, it's doing the violin plots. It's over here. Let me just verify plots, and then we can do violin plot, and we're gonna compare the batches, and as they are, it's fine. Where is my friend? Gene expression, and I'm gonna paste the same. Actually, now I can paste all the set we got in the overlap. We can just come back and copy all these guys and paste that list here, and we can verify the expression level, normalizing A and B, and it's gonna plot all the genes in the same figure. Probably not for this case. It sometimes helps for publication, but in this case, we are gonna just look one by one. Okay, this is exactly what we were looking. That is upregulated in the 14 weeks and wiggles. That means variant. This other also wiggles. Perfect, it's variant. The gene we were talking about, CLCX, okay, it doesn't look like wiggling. Why? Because it was strongly expressed before in the 14 week than in the 8 week. So this is powerful enough to tell, hey, things are changing and are unstable, and that instability can be natural or can be disruptive by some disease. That's something we've been promoting with this method. And so far, this is all I can offer for a spline-DV method and how to interpret the differential expressed and differential variant genes. Differential variability has one weakness, and is that it does not have directionality, that all the people want to know if it is upregulated or downregulated. But those are powerful enough to tell that something is broken. If you have a healthy tissue and you compare it to the cancer, definitely if you have a variant gene in the healthy, that should be normal. And if the variant is decreased in the cancer, that means that cancer is stabilizing that for some reason. And so far, I think that's what I can offer. And I hope you enjoyed this. It's not easy, it's iterative, and has to be repeated several times. And I hope you learn more while you practice. With this, I just say bye-bye. Thank you for watching.